Welcome. Today I want to talk about the hybrid technology and advanced indoor tanning. If we skip back, let's say 15 years ago, what the customers or the users expected when they went to a tanning salon, when they used a tanning bed? It was the maximal tan. And maximal tan can be achieved with mainly UVA radiation. Other parts of the spectrum didn't really play a significant role these days. So let's skip back just 10 years into the past. Something already changed. Users expected more than just the maximal tan. They were thinking about other effects, about the tan and more. And more, these are the so-called biopositive effects. As an example, as an important example, we are talking about vitamin D here, the photosynthesized vitamin D, which is directly produced in the skin, in the outmost layers of the skin, and which is transported in a different way compared to orally taken vitamin D. So how to achieve the photosynthesis of vitamin D? You cannot achieve this just using UVA alone. You need another part of the spectrum. You need UVB. UVB between 290 and 320 nanometers. And to give you a better impression where we are in the spectrum, I'll show you the spectral ranges which are important for our cosmetic applications and more. Here we see the sunlight and we see different electrical light sources. But we can forget about 50% of this slide, because everything which is painted in red just causes kind of thermal effects, and we are talking about other effects, more specific photobiological effects than the thermal. So let's focus in to this and make it a little bit larger. Here you can see again the sunlight in the upper row, and then all the other light sources from incandescent to the fluorescent lamps we are using actually for producing the spectrum, the ultraviolet spectrum. And what you can see here in the blue box is the standard spectrum of a tanning booth. And if we compare this small spectral region of the tanning booth with natural sunlight, we can easily see that we have much more different wavelengths contained in the sunlight itself. So this is our blueprint, and if I would make a comparison, I could say using just this small part of the spectrum, this small segment, it is comparable to playing just a simple flute, one single instrument. And what sunlight in fact does, it's comparable to the full sound of a philharmonic orchestra. And we have all the different instruments in sunlight, and one principle of the photobiology in the 20th century was to focus just on several small parts of the spectrum where the photobiologists thought they could achieve the maximum effect. As an example, maximum tan or maximal production of vitamin D. But if you only have a small part of the spectrum, could be compared with the situation that you only have a hammer. In this case, each problem looks like a nail. And if we only focus on these small segments of the spectrum, we have to say goodbye to all the different positive effects. We have to say goodbye to the full bunch of biopositive effects we could achieve if we would add some additional parts of the spectrum with beneficial effects. And this, to focus on just the small part of the spectrum, this is pretty much 20th century. We are now in the 21st century, and we can expect much more from the technology from the equipment of a modern, recent, contemporary tanning booth. And what the customers today expect? They are much more aware of the health effects. They seek for wellness and beauty. And if we can offer them a softer tanning, this is much, much better, because compared to the narrow band maximum tan, the softer tanning, it just feels pleasant. You have a feel-good skin, and the wellness starts in the outmost layer of your system. The wellness, the feeling good, starts in your skin. And how can we achieve this? Let's skip back one more time to the spectral ranges for cosmetic applications. You already know this chart. And here you can see the extended spectrum. This is the second row from the basis. You can see we have more than just the UV. We added the so-called collagen spectrum in the red part, in the longer wavelength part of the spectrum. And what you can also see on this chart, this is the very narrow bandwidth red, which is produced by a laser. And the laser brings maximum energy, but it's not feasible for a tanning booth. We see also the LED spectrum, and this is what we can achieve nowadays, to put more than just one light source into one unit, into one tanning booth. But now something a little bit different. Let's talk about the skin. Let's talk about what is a beautiful skin. Beautiful skin signals health. Beautiful skin is a very important code in human communication, and our eye is very sensitive to the very specific use the skin can take and the skin can offer. So it's a kind of projection area of health, of wellness, of well-being, which is visible to all the others around us. But if we go a little bit more into the details, how the color, how the hue of the skin, the healthy appearance of the skin is composed of, it's not only the tanning mechanism, it's not only the melanin, which is found in the upper layer, in the epidermis, because the melanin only gives a gray value, and we need, for a brown or for healthy skin, we do not only need the gray values, we also need the red values. And the blend of the perfect value in the red and the perfect value in the gray, this gives the beautiful skin color. Too much gray does not look healthy, and too much red doesn't look healthy either. If we are thinking about the UV only, you can see in this chart, the sunlight, the UV from the sunlight only reaches the outer layer of the skin, only reaches the epidermis. But if we want to influence the blood circulation and the reflection from the capillary layers in the dermis, we need other parts of the spectrum. We need some spectral ranges which are able to penetrate deeply into the tissue and which are able to influence the metabolism and the blood circulation in these deeper layers of the dermis. And here I have another picture which makes it hopefully clearer. The short wavelengths of the radiation reach only the leaves, which can be compared with the epidermis. And the long wavelengths, the red, reaches the roots. And you all know, if you have a garden, for example, you know that you only have healthy plants when you feed the roots with nourishing substances and water, and when you supply the light to the leaves. So addressing both layers, the epidermal layer and the dermal layer, we not only should use the short wavelength part. In addition to the ultraviolet radiation, we can use the red light, because this penetrates much, much deeper into the skin. And to make this even clearer, I have a representative for the short wavelengths, blue light, and the representative for the long wavelengths, the red light. And when I put it on my fingertip, you can see there is almost no penetration for the blue light, but a significant penetration for the red light. So this we can use to nourish the leaves and to address the leaves, and this we can use to address the roots. So if we make another experiment with these little light sources, they represent both ends of the spectrum. The red light represents normally the warm end of the spectrum, and the blue light represents the short wavelength, the cool part of the spectrum. But if we do the reality test, if it would happen that you have these two little LEDs, you could feel on your lip that there is almost no thermal reaction, no heat to feel when you shine it on your skin for the red light. But for the blue light, you immediately feel that the tissue warms up. It starts heating up. And if I would have some violet light, it would be even more intense. And this intensity can reach a certain level where it even becomes uncomfortable, and it turns into kind of very sharp heat, into a sharp thermal effect. And this leads me to the next topic, and this topic is what happens under the influence of radiation in the skin. It depends on the wavelength, and the sharpness of the thermal effects of the short wavelengths mainly come from high absorption as one aspect, and from the production, the immediate production of free radicals, or so-called reactive oxygen species. Reactive oxygen species, you can look at them as doorkeepers. They can be very, very helpful. Our organism needs these molecules for defensive effects, for example, for destroying germs, bacteria and viruses in the skin, and as very important signaling molecules which influence the energy metabolism, for example. And almost all the effects, all the light reactions, like the pigmentation, like the production of nitric oxide, which is responsible for the microcirculation increase in the capillary layers, which is responsible, for example, for lowering the blood pressure. All these light-induced effects come from the production of reactive oxygen species. In the right concentration, they are very helpful, and we couldn't live without them. But if they exceed a certain level, they become harmful and dangerous. So the doorkeepers really turn into free radicals, and they can become very dangerous for our body cells, for the skin cells, if they appear in too high concentrations. So the best effects, let's say it's all about the optimal blend. The optimal blend, like we find it in sunlight. Sunlight is our blueprint for radiation type, which we are accommodated for since many, many thousands and thousands of years. So let's go a little bit deeper into this topic. The desired effects of the short wavelengths, for example, vitamin D production in the skin, and the right dose of reactive oxygen species. They are signals for building the horny layer of the skin, for inducing the pigmentation. So all the photoadaptation mechanisms are induced by reactive oxygen species. But if we increase the dose, we have not only the desired effects, we can also have negative effects, in fact, cellular stress. For example, a sunburn, this is an overdosage of reactive oxygen species. It's a kind of ROS intoxication. On the other hand of the spectrum, we find the opposite of cellular stress. We find cellular regeneration and repair. And What are the mechanisms here? The red light stimulates some biomolecules in the powerhouses of the cells, the so-called mitochondria. And the stimulation of mitochondria via red light results in the increased production of ATP. ATP is adenosine triphosphate. This is the fuel our body cells live from. So no energy consumptive process in our body would work without ATP. So repair is increased activity, and it just works better with more ATP. And another positive effect of this long wavelength range in the spectrum is the production of additional antioxidants. Antioxidants are molecules which compensate for excessive concentrations of reactive oxygen species. So this is the antitoxin, so to say. And as we can tell, as you might tell, our body, our cells know exactly about the right dose of antioxidants. So everything you ingest orally cannot work in the same efficient way than those antioxidants produced on-site in the cells. And we learned that red light has these positive effects, but there is a broader range of red light. Why do we use exactly this wavelength in our latest equipment? Why do we use LEDs with a wavelength of 628 nanometers? Because this is the safest part in this spectral range. This is the frequency. Almost all the research with photobiomodulation, with non-thermal light effects, had been performed. So it's safe, and we can look back over 50 decades of scientific experiments with this specific wavelength range. It started with the neon-helium laser, which has exactly 632.8 nanometers, but the cells, they don't care. It's more or less the same if you have 620 or 640. Somewhere in between we have the maximum effects. The researcher, the Estonian researcher, Tiina Karu, for example, she made hundreds and hundreds of experiments and published tons of papers, of scientific papers, proving that the spectral range between 600 and 640 nanometers is optimal for a maximal ATP production, for a maximal cell fuel energy production. And the majority of the studies comprise this wavelength range. For example, our belt study also had been performed using light sources which produce the 630 nanometer. So let's have an even deeper look, like kind of summary of this part of the presentation. Here we see the reactive oxygen species depending on the wavelength of the radiation. And in the blue circle you can see all the free radicals which can be produced by ultraviolet radiation, UVA, UVB, and also the short wavelength part. This is what I demonstrated with the little LED. This is in the visible part, the activity of creating these ROS molecules. But also in the long wavelength part, here on this slide, in the red circle, you see the green molecule. This is the cytochrome C oxidase molecule. This is the important molecule which is addressed in the mitochondria, in the cellular powerhouses. And if you shine red light onto this cytochrome C oxidase, there is an increase of ATP. And there is also, in a second phase, an increase in antioxidant production in the cell. This mechanism is called mitochondrial signaling. Increased reactive oxygen species in the mitochondria signal to the nucleus of the cell that there is a need for antioxidants. So the consequence of all what I told you up to now is that it's really a good idea to combine the short parts, short wavelength parts of the spectrum with the long wavelength parts. We address the upper layers and we address the deeper layers. We take care for the correct concentration, so we have the positive, biopositive stimulating effects from ROS molecules. But on the other hand, using the red light, we take care that there is no exceeding concentration of ROS, so that they're in the optimal level range. So this is what I told you up to now, and now let's have a short look into science. What can we learn from science? There were some hundreds of interesting papers published in the last years, and I just picked out some significant papers to bolster what I told you. This is a study which can demonstrate that with red light you can protect the skin cells, in this example fibroblasts in the dermis, from deleterious effects of solar UV radiation, so it has a protective effect. The next study demonstrates that red light enhances the cellular resistance to ultraviolet radiation, and this sounds quite theoretical, but here is a slide which demonstrates what is meant by this. On the right side you can see a stepwise increase of ultraviolet radiation doses, and you see the little square areas which had been addressed with ultraviolet light, and you have a reddishness, a stronger reddishness on the red side compared to the left side, or in other words, the left side of this back of a patient was more resistant to ultraviolet radiation compared to the right side. More reddishness, more erythema on the right side. And what is the reason for that? On the next slide we remove the bar, and you can see the left side had been pre-treated with LED light, with red LED light, which made the skin much more resistant to the ultraviolet radiation. So even again here, it's a good idea to combine the red light, the long wavelengths with the short wavelengths. And one last paper demonstrates that we have another pathway induced or stimulated via red light for the cellular production of antioxidants. So this is enough science for today. I want to summarize again. Our research focus is to combine the short wavelengths with the long wavelengths in terms of achieving a balanced spectral distribution for optimal effects. This was it. Thank you very much.